Hi, my name is Julia Oliveri, and today I'll be telling you about statistical detection of regulated alternative splicing from single-cell RNA sequencing data. Single-cell regulation of alternative splicing is functionally essential, but is currently not well studied. A few interesting alternative splicing examples that have been extensively studied at the single-cell level have mainly been in the brain. For example, the transmembrane protein DSCAM expresses over 38,000 isoforms in the Drosophila brain. This is enough so that each neuron can express a different set of DSCAM isoforms, which act as a fingerprint so the neuron doesn't synapse to itself. Alternative splicing has been massively understudied at the single-cell level, especially in non-brain tissues, meaning that there are critical unanswered questions about its functions in all cells. Single-cell sequencing provides a new opportunity to discover cell-type-specific function and regulation of alternative splicing by reporting the cell of origin of each RNA molecule, as opposed to bulk RNA sequencing, which doesn't differentiate between RNA molecules from different cells. Over the course of the rest of the talk, I'll tell you about challenges of studying alternative splicing in single-cell RNA-seq, the new statistical method we've developed to identify single-cell regulated alternative splicing, and some of the new biological discoveries this method has enabled. It's currently an assumption in the field that alternative splicing data from single-cell RNA sequencing is too sparse for statistical inference, especially data from droplet-based platforms. Our data looks kind of like this, with very few splice junctions present for each gene in each cell. With data this sparse, can we draw any conclusions? We can back up for a second to think about the question under deeper sequencing. Although the answer still isn't obvious, we can use statistical methods to differentiate these cell populations. But as the data gets sparser, we need more and more rigorous statistical methods. Naive methods can lead to misleading conclusions. If we only ever have one measurement per cell, we could conclude that each cell only ever expresses one isoform. However, because we have thousands of cells in single-cell RNA sequencing experiments, using the appropriate methodology, we can draw robust conclusions even under sparse sampling. The approach we've chosen to take to answer this question is called the Splicing Z Score, which captures the alternative splicing of each gene in each cell with a single score. The basic intuition of the SES is that for a given gene, we want to find whether the introns, which are the parts of the RNA sequence that are spliced out, longer or shorter than average. So for example, for this cell, this gene would get a low SCS value because the introns are short, whereas this cell would get a high SCS value because the introns are long. Some benefits of this method are that the calculation is efficient, it can capture complex alternative splicing events as well as simple exon skipping. We've developed a new method to calculate the false discovery rate, which gives us more confidence in the reproducibility of our results. And we get one score per gene per cell, which result in normal scores with mean 0 under the null hypothesis, making them easy to plug into downstream methods. Because alignment of spliced RNA sequencing reads is known to have a high false positive rate, we use Sicilian to prepare our data. Sicilian is a tool developed by the Saltzman lab to filter low-confidence spliced reads from star alignments, allowing high-confidence unannotated splicing to be reliably discovered. The SES can be used for metadata-guided differential splicing, alternative splicing detection. Because the SES is normally distributed with mean 0 under the null distribution, we can look for cell types with median SES values far from 0 to identify differential alternative splicing. After we developed this method, we wanted to discover, does it actually capture new and interesting biology? To answer this question, we analyzed hundreds of thousands of cells sequenced using the 10x platform from the Tabula sapiens cell atlas. When we applied the SES method blindly to the Tabula sapiens dataset, we identified multiple known true positive genes, including the myosin regulating gene myl6, which was the gene that was differentially alternatively spliced in the greatest number of cell types. myl6 expresses two isoforms, which differ by an exon skipping event. Looking at this donor, we can see that the splice junction between the donor and acceptor 1 indicates that the isoform for which the exon is included, whereas the junction between the donor and acceptor 2 indicates the isoform where the exon is skipped. This alternative splicing switches the 9 amino acids in the alpha helix at the end of the protein C-terminus. Here, I'm showing for one given cell type the fraction of junctions with this donor that spliced to acceptor 1 versus the fraction that spliced to acceptor 2. And these two dots represent the 2 biological replicates of 2 different individuals. We can see that in this cell type, smooth muscle cells, about half of the isoforms include the exon and half don't include the exon, which is what was known from the literature. However, when we analyze a single-cell RNA sequencing dataset with the SES, we get information about this gene for all cell types, allowing us to also uncover new biology beyond just known true positives. For example, immune cells, shown here in green, almost exclusively skip the exon, whereas epithelial cells in orange mostly skip the exon, and endothelial cells in blue express the skipped and included versions about equally, similar to the stromal compartment in red. This is just one example of the hundreds of genes that we found to be differentially alternatively expressed in this dataset. We found 219 genes to be differentially alternatively spliced across 153 cell types. The genes that were differentially alternatively spliced in the most cell types were MYL6, RPS24, and RPL27A. An example of new splicing biology we found was in the lineage-specific splicing of CD47, which is a critical cancer immunology target. CD47 has many different annotated isoforms, and alternative splicing of these exons affects the plasma tail of the protein. We can see that this acceptor has 4 different donors expressed in this dataset, and there are strong compartment-specific effects of splicing in this gene. For example, stromal and immune cell types tend to mostly just express the junction between this acceptor and donor 3, whereas epithelial cell types have a more uniform distribution across the 4 donors, preferentially using donor 1 and donor 2. This is an example of how the SCS can discover complex alternative splicing patterns beyond just simple exon skipping. Because we can calculate the SCS for each gene in each cell, if we have cell-specific metadata for our dataset, we can perform correlations with the SCS. We investigated this for spermatogenesis, which is a highly regulated developmental process. We analyzed around 5,000 cells from one mouse lemur from the Tabulum microcebus atlas. We correlated the SCS for each of the genes found in these cells with pseudotime measurements of the developmental trajectory, and we found 287 genes with significant Spearman correlations. 4 of the top 8 correlated genes are known to be associated with testes function, which indicates that splicing here is likely to reflect functional changes. The testes-related gene TEX43 had the most significant correlation between the SCS and the developmental trajectory. This acceptor has 4 different donors in the dataset, and we can see that immature sperm tend to use donor 4, transitioning slowly to mature sperm, which mostly use donor 2. Sicilian allowed us to identify the splice junction with donor 4, which is unannotated. The SCS opens the door to alternative splicing analysis in single-cell RNA sequencing data. Alternative splicing has been massively understudied in single-cell RNA sequencing datasets, especially droplet-based data, but the SCS enables us to identify a wide range of biological regulation of splicing, everything from clustering to alternative splicing in development or the cell cycle, or regulation with transacting factors. Finally, I would like to thank my advisor, Julia Saltzman, and Rusbeta Ganasari and Peter Wang, which have been the main other contributors to this work. I'd also like to thank our collaborators, especially the Tabula Sapiens and Tabula Microcebus teams. And As always, I'm thankful for my lab members, and if you're interested in this work, we have a preprint about the general method available on bioRxiv. Thank you.